Okay, so we're going to go ahead and look at membrane transport, and we're going to talk about how do molecules go from one side of the membrane to another, and what's the driving force of that. So there's kind of two types of membrane transport. The first type is passive transport, which requires no energy from the cell. And then the other type is active transport, which you can see is requires energy from the cell. Usually in the form of ATP. Usually in the form of ATP, as we know is our energy currency of our cells, okay? So usually if you see that it's using ATP, it's active transport, and if it's not using ATP, it's passive transport. It's kind of a general rule of thumb. So how does the molecule then get across a membrane, or what drives it? We use concentration gradients. So molecules want to move from high concentrations to low concentrations. They want to go from where that's really crowded and there's a lot of them to where there's less of them. So they want to diffuse across a membrane if possible, or somehow move from where there's a lot of them to where there's not so many of them, whether that's like a sodium molecule, a water molecule, a potassium molecule, anything, a glucose molecule. They're going to go from where there's highly concentrated, like in the bloodstream, into the cell. Now they might need help, which we're going to talk about how they can get help to go across the membrane, because they're large or polar or something like that. But they, generally speaking, the energy source, the quote unquote energy source, is just moving from a high concentration to a low concentration. So if they're doing that, it's usually passive transport. If they're going against their concentration gradient, so let's write this. If molecules go against, so they're going from low to high, then it requires energy of some sort. Okay, and then the last part that goes up with the second learning objective, how do we organisms that use to like transport large molecules across the plasma membrane, if they're trying to get something from the outside, a large amount of a substance. So here's my cell. I'm going to kind of draw it in stages. I'm drawing endocytosis here. And then just outside my cell, I have a bunch of molecules that I want to take in. Let's just say that's a bunch of starch. I don't know. And then, so what's going to happen is my cell is going to begin to wrap around it, if you will, and engulf it, or endocytose it, bring it to the inside. So it begins to wrap around it, and then here we go. And eventually, we're going to have our cell, and I'm going to leave this like this. Then we're going to have like a little vacuole, a little pinched in part of the membrane that has a bunch of that molecule from the outside. Okay, so it's actually going to pinch in. So that's endocytosis. And then exocytosis, let's do a different color. Let's say I want to get rid of a bunch of molecules. Maybe I'm trying to release a bunch of hormones. Like I'm trying to release a bunch of insulin that I made out into the cell. So in that case, it's just really the opposite process, right? So there's my vacuole filled with my molecules. In this case, they're going to be that lime green color. And so then that vacuole is going to end up really close to the membrane. I changed blues, but that's okay. And it starts to kind of come out like that. So there's my vacuole. And my molecules. And then eventually, back to the regular blue. My cell's back to normal, and my molecule that I made is out in the extracellular matrix, maybe in the bloodstream, going out, something like that. So exocytosis is where I release chemicals out or a molecule out, and endocytosis is where I engulf them. And they involve vacuoles, which we'll talk about when we get into the cells part of this unit. Okay, So that's just some of the ways that we transport things across the membranes and the mechanisms for it.